Well, it's a great honor to be asked to give the 2019 Leeuwenhoek lecture. And I'm grateful to the nominations committee and the Royal Society, but most of all to the people who've worked in my lab over the last 30 years or so who've made this award possible. So very many thanks to you all. So this is the department I work in Cambridge, and here's my contact details should you want to contact me after this. On this slide, I'm showing the plan for the lecture. A few words about Anthony van Leeuwenhoek, and then talking about the eradication of smallpox, how this was achieved, and then the post-eradication issues, when the disease had been eradicated, but the virus remained, and what to do with it. And then I'll finish by talking about some new findings working either with the genome of variola virus with ancient DNA and looking at the virus evolution from those ancient DNA samples. And finally, talking about how this group of viruses, the pox viruses, are really expert at evading the innate immune system. And the example I'm going to give is how vaccinia virus interferes with interferon. So van Leeuwenhoek was a Dutch draper. He came from Delft and he developed microscopes. And it was that made him famous because with them, he was able to discover bacteria. And he described most accurately the structures of bacteria, the cocci, the rods, and their sizes, and also their motility. And the accuracy of his measurements have really been astonishing and have been confirmed by more modern investigations of bacterial size and structure. Another important thing that he did was to demonstrate the existence of unicellular life. And this was a concept that was not accepted until this demonstration and was received with skepticism by the Royal Society. Indeed, the Royal Society dispatched a delegation to Delft to visit van Leeuwenhoek and to look at his data. Well, they were convinced. And just 3 years later, he was elected a Fellow of the Royal Society in 1680. But he neither attended that ceremony, nor indeed any other Royal Society meeting. So perhaps it's fitting then that this lecture to honor him is also given without a formal get-together. Well, he's been described rightly as the father of microbiology because he discovered bacteria and he developed these wonderful microscopes. But even those microscopes were not powerful enough to detect another group of microbial pathogens, the viruses. And these are much smaller and indeed were not discovered until the end of the 19th century when first Ivanovsky and then Loeffler identified pathogens of plants and animals that were able to pass through the filters that would retain all known bacteria and therefore were very small. And these are viruses. And this lecture is going to talk about one group of viruses, in particular, the pox viruses. These are large double-stranded DNA viruses that replicate in the cytoplasm. And the most famous one, or infamous one, was variola virus that was the cause of smallpox. And this is a now extinct human disease. And this WHO identification card illustrates an important feature of smallpox, which helped distinguish this disease from other diseases like chickenpox that are caused by a herpes virus. And on the face of this poor child infected with variola virus, you can see that there are abundant lesions. And these are much fewer on the trunk, at least early in the disease. And this so-called centrifugal distribution helped distinguish smallpox from chickenpox. Well, variola virus came in two types, variola major, that had a very high mortality rate associated with it. In unvaccinated populations, the average was about 30 to 40%, but it could be higher than that. And with variola minor, only 1 to 2%. But lest we think this is a low figure, we should remember that the great flu epidemic of 1918-19 that killed 50 million people had a mortality rate of only 2.5%. And the current SARS coronavirus 2 pandemic has a mortality rate which is similar to this and has already killed 5 million people. While smallpox has played important roles in human history, it was a devastating disease that, like measles, required large populations to persist. And so this would only have happened in man when we changed from being hunter-gatherers to city dwellers and farmers so that large populations could build up. And that would have only happened perhaps 5 to 10,000 years BC. So smallpox has profoundly influenced human history. And one example of that is with the conquest of the Aztec nation by the Spaniards in the early 16th century. So unwittingly, the European settlers took to America pathogens from Europe that had not been seen in the Americas hitherto, such as measles and smallpox. And it was a devastating epidemic of smallpox in the Aztec nation, which persuaded them to give up as much as the onslaught from Señor Cortés and his 500 Spanish soldiers. Later that same century, our first Queen Elizabeth, just 4 years into her reign in 1562, became gravely ill with smallpox. But fortunately, she survived and, of course, went on to be a long-lived and a strong European monarch. In North America, about 300 years later, Abraham Lincoln became infected with variola virus. And you may not know this, but when he gave his most famous speech, the Gettysburg Address in 1863, he was suffering from smallpox. So how remarkable to be able to give such a magnificent speech and to do it while suffering from smallpox. Well, in the 20th century alone, this terrible disease killed an estimated 350 million people. And this occurred more than 100 years after a vaccine and an effective vaccine was available to prevent it. Well, how was smallpox contained? Well, many of you will know the story of how Edward Jenner noticed that the milkmaids, who sometimes became infected on their hands with cowpox from the cows they were milking, milkmaids were somehow spared the ravages of smallpox when an epidemic would rage through the community and seemingly everyone else would be afflicted. And although Jenner was not the first person to notice this correlation, he deserves the accolade for the discovery of vaccination because it was he who set out to test experimentally the hypothesis that it was the prior infection with cowpox that was the cause of resistance or protection against smallpox. And so he did what by today's standards would be a totally unethical experiment. He would probably be jailed. And he took his gardener's son, a boy called James Fitz, and he infected him with cowpox material taken from the hand of a milkmaid named Sarah Nelms. And as if that weren't bad enough, a few weeks later, he challenged the child with smallpox. And the child resisted the challenge, and so Jenner wrote up his paper and submitted it to the Royal Society, of which he was already a fellow for his work on ornithology. The Royal Society, rather as they had shown skepticism to Leeuwenhoek, rejected Jenner's paper, telling him he was unwise to risk his good reputation on such an idea. And so rather insulted, Jenner went away, collected more data, and then published his paper privately in 1798. Well, it was blindingly obvious that vaccination worked, and this enabled Jenner just 5 years later to predict the eradication of smallpox. And in 1801, he wrote, "It now becomes too manifest to admit of controversy that the annihilation of the smallpox, the most dreadful scourge of the human species, must be the final result of this practice." And he was right, but it did take another 176 years. So why did it take so long? So we've dealt with the introduction of vaccination and the prediction of eradication. And during the 19th century, the vaccine had to be spread around the world. And smallpox is now, and was then, a rare disease. Cowpox is a rare disease. And so the vaccine was not available throughout the world where it was needed. And this was spread in part by arm-to-arm transfer. So for instance, the Spanish, to get the vaccine from Europe to their colonies in Central and South America, used children and put them on a sailing ship and vaccinated them at different cohorts of them at weekly intervals across the Atlantic. This practice of arm-to-arm transfer, although effective at transmitting the smallpox vaccine, could also enable transmission of other diseases such as measles or syphilis if the vaccinee was infected with those diseases. And so eventually this practice of arm-to-arm transfer was banned. And indeed in UK, it was banned by an Act of Parliament in the 1890s. Well, in 1939, Britain became free of smallpox for the first time. And in that same year, Alan Downey, working in Liverpool, showed that all the smallpox vaccines used in the 20th century were not cowpox. They were a distinct virus that became known as vaccinia. And the origin of vaccinia remains an enigma of virology. In the 1950s, Leslie Collier, working at the Lister Institute in London, developed a way to freeze-dry the vaccine so that now it could be transported around the world without the need for refrigeration. And this is a massive advantage for any vaccine and was a very important development for the global eradication of smallpox. In 1959, the World Health Assembly adopted a proposal put forward by the Soviet Union to achieve the global eradication of smallpox. And this was enthusiastically endorsed. But in 1967, the policy changed because little progress had been made. And now they used ring vaccination, where they would identify cases of smallpox by widely distributing the smallpox identification card I showed earlier. When a case was identified, the patient would be quarantined and the contacts traced and vaccinated. And in this way, a ring of immune people was thrown around the site of infection. And this was a very effective way of containing and eliminating the virus. So the last case of smallpox in the Indian subcontinent was in 1975, and the last natural occurring case anywhere in the world was in Somalia 2 years later. After a further 3 years of extensive surveillance, the WHO certified that eradication was complete in 1980. And 13 years after that, the genome of the variola virus was sequenced. Note the order. So smallpox is the only human viral disease to be eradicated. And the estimated cost of this was just $250 million. So why was smallpox eradication possible? Here are the lessons for the control of other diseases. So the first thing was that variola virus had become a virus that could only infect man. There was no animal reservoir. And so once the virus was removed from man, it couldn't be reintroduced from other sources. Contrast that with the situation with rabies or with yellow fever that have many mammalian or insect vectors. The nature of the infection itself was also an acute infection. If infected, you became ill and you either died or you recovered from infection and cleared the virus from the body. There was no latency or persistent infection as we have with herpes viruses, for instance, which many of us will carry for the rest of our lives. So this was not a problem with smallpox. It's also an easily recognizable disease. If you saw a child looking like this, you could not fail to notice, and therefore action would be taken. And we could contrast this with the tragedy of the HIV epidemic, where when the first cases of AIDS were recognized in Los Angeles in 1981, the virus HIV had been spreading in the human population for years and indeed decades. So the epidemic was out of control before we knew it existed. And the vaccine produced was effective against all strains of variola virus. Variola was not able to undergo antigenic variation like influenza or hepatitis C do. and therefore it could not escape the immunity from prior infection or from vaccination. And lastly, the vaccine had many properties that made it ideal. It was potent as a single dose, it was low cost, it was abundant, it was heat stable when freeze dried, and very easy to administer under field conditions. And lastly, we should not overlook the real determination of the WHO to see this through. And it's a great example of how, where nations work together, they can achieve something of mutual benefit. Well, why was smallpox vaccination effective? There really was no understanding of this until after eradication was complete. When the proteins from the outer envelope and capsid proteins of vaccinia virus were identified and compared with the corresponding ones of variola virus, they were shown to be very highly conserved. And by that, I mean greater than 90% amino acid identity. So these facts were only discovered long after eradication. And the lesson from that is to have an effective vaccine and indeed use it to achieve global eradication, you don't need to understand very much about how it works. And so if you have a vaccine that works, we should use it. And I really mean that for MMR, of course, which is being underutilized, and indeed the COVID-19 vaccines, which are available today. It's tragic that 60 years after the measles vaccine was first produced, measles are still with us. And indeed, in 2019, there were more than 200,000 deaths worldwide from this entirely preventable disease. So now to the post-eradication issues. So smallpox had been eradicated, but the virus that caused that disease remained. Vaccination had stopped, and so an increasing proportion of the population was now susceptible to smallpox. So the WHO first catalogued and collected and centralized all the known virus stocks in laboratories in the United States and in Russia. And then a debate was held about what to do with the virus remaining. Should it be destroyed or retained? And those advocating destruction argued that it's the logical next step after eradication. Why keep it? It removes all the risk of the virus escaping, and it removes the need to store the virus safely and securely. And those advocating retention, at least for the moment, would argue that the virus would be needed to test safer vaccines and antiviral drugs and diagnostic tests in the event that smallpox ever re-emerged. And also understanding why the virus was so virulent and able to overcome our immune system would be useful for our understanding of viral pathogenesis that could be applied to other viral diseases also. Members of the Green Party might have argued that we should not destroy biological diversity. Well, the post-eradication policies of the WHO were overseen by a committee on orthopox viruses that met in the 1980s regularly, and then 3 times in the 1990s. And at their meeting in 1994, the committee debated whether the remaining viruses should be retained or destroyed, and recommended that they should all be destroyed. And this recommendation was debated and endorsed by the World Health Assembly in 1996, and a date for destruction was set for 3 years later. 22 years after that, the viruses are still there. So what happened? Well, in 1999, a delay in destruction was authorized to enable essential research for public health benefit at these 2 WHO collaborating centers to be completed. And the objectives were to produce diagnostic tests, antiviral drugs, and a safer vaccine for those who were contraindicated. But the challenge with this program was, how do you test drugs and vaccines against an extinct human disease? The Federal Drug Administration or the Medicines Control Agency, to license a drug or a vaccine, would require evidence that it works against its natural pathogen in the natural host. And of course, we can't do that with smallpox in man anymore. So to oversee the research, the WHO set up this committee called the Advisory Committee on Variola Virus Research that was convened to oversee the work with variola virus. And it's met annually for the last 20 years or so. And each year it will approve or disapprove research proposals that must have specific objectives and be time limited. And then after each meeting, a report's produced, goes to the Director General, to the WHO Executive Committee, and then is debated at the World Health Assembly. And these reports are in the public domain. You can all read them. So what is the progress towards those objectives? Well, it's really been terrific. And indeed, diagnostic tests have been developed based on PCR to detect the viral genome. These are sensitive and specific. Safer vaccines have been developed, and indeed modified vaccinia ankara, an attenuated form of vaccinia, has been licensed for smallpox vaccination, both in Europe and in North America. And new compounds, drugs that work against orthopoxviruses, including variola virus, have been produced, and they're called Tembexa or TPOXX. And TPOXX was licensed by the US FDA in 2008, and the US government placed an order shortly thereafter. And this was remarkable. It had several firsts. It was the first drug that had ever been produced for smallpox, because there were none during the eradication campaign. It was the first time a drug had been produced and licensed against an extinct disease. Why would you? And it was the first time that a drug for use in humans had been licensed based upon efficacy data that derived only from animals. Well, this year, a second drug, Tembexa, was licensed, and so there are now two drugs that act at different places in the virus life cycle. And that's illustrated in this slide, where once the virus infects a cell, it sets up a factory in the cytoplasm in which new viral particles are produced, and then these are transported out of the factory. They become wrapped by additional membranes and then are transported out of the cell. So Tembexa works by targeting the viral DNA polymerase and so blocks replication and no new virus is produced. TPOXX, however, doesn't block replication, but it blocks this wrapping stage. And so the virus cannot be transported out of the cell and its dissemination is restricted hugely. And despite not blocking replication, this is an effective drug so long as there is an effective immune system present to remove the virus-infected cells. So to some degree, the debate about whether to destroy variola viruses has been influenced by developments in synthetic biology. And in just 3 years ago, it was demonstrated that pox viruses could be recreated from chemicals. And this was done by Noyce et al. Using horsepox virus, a strain of vaccinia. And what they did was to synthesize the horsepox virus genome in approximately 20 kb chunks that were slightly overlapping with each other, and the terminal hairpins from both ends, and introduce these into cells, which were then infected by a helper virus that would provide the enzymes that could transcribe the genes within these DNA fragments. And the proteins then produced would enable the different parts of the genome to assemble by homologous recombination, and the fully assembled genome would then be replicated and packaged into new viral particles. And these would be released from the cell, and they could be distinguished from the helper virus by the growth of this virus on a cell line that was permissive for the recreated virus, but non-permissive for the helper virus. So this was done, and it's been done for horsepox, and so it can be done for other orthopox viruses also, including variola virus. Though, of course, this is absolutely prohibited. But nonetheless, because this is theoretically possible, it is always possible that someone might recreate variola virus, and therefore the threat of smallpox can never be eradicated. So for the last few minutes, I want to talk about variola virus and its genome and what's been learned about that in the last few years. So broadly, if you look at an orthopox virus genome, it's a linear double-stranded DNA molecule with terminal hairpins that encodes about 200 genes. And the central region of the genome is very conserved between different strains of variola and indeed different orthopox viruses and caudopox viruses. And the genes encoded in this region are essential for virus replication. They encode the enzymes for DNA and RNA synthesis and for assembling new virions. Whereas the genes present at the terminal regions of the genome are more variable, and these are mostly non-essential for viral replication in cell culture, but they affect the virulence of the virus and in particular, the strategies for immune evasion. And it's estimated that between 1/3 and 1/2 of all the genes encoded by orthopox viruses are dedicated to immune evasion. So these, if you like, this collection of proteins are a Brazilian rainforest of molecules that have been evolved to target specific aspects of our immune system. And studying how they work is both fascinating, but also provides fundamental information about how our immune system works. So when the first pieces of variola virus were sequenced and compared with vaccinia virus, there was a surprise. So this was a paper from our lab when I was in Oxford in the 1990s. And a region of variola virus strain Harvey was sequenced, a 22 kb piece from near the right end. And this was compared with the corresponding region of 2 strains of vaccinia, WR and Copenhagen, that had been sequenced in our lab and by Enzo Paretti's lab. And you can see that the genes of variola on the top line are pretty much collinear with those of vaccinia, at least in this region. But what was surprising was that in multiple places, a gene which was intact in the vaccinias was broken by multiple mutations in variola virus. So these genes are smashed to bits, and there's no way that they could encode a functional protein. So that was really surprising. And over the last 20 or 30 years, the functions of many of these proteins have been deduced. And in summary, what these proteins do, at least in the vaccinias and the other viruses that make these proteins, is that they suppress the immune system. So this one is an enzyme that synthesizes steroid hormones. These two block NF-kappa B activation. This one's involved in evading recognition of infected cells by NK cells. And yet all these are broken and non-functional in variola. And intuitively, this was sort of back to front. You might have expected the real pathogen to have its genes intact and the vaccine that prevented that terrible disease to have the genes disrupted. Not so, it's the other way around. But presumably, these viruses both derive from a common ancestor which had the genes intact. So when did the genes become fragmented in variola virus in its evolution? Well, clues about that came from studying variola genomes obtained by sequencing ancient DNA. And the first example came from the mummified body, the mummy of a child who died in about 1650 from a crypt in a church in Vilnius in Lithuania. And mass sequencing of DNA from that mummy showed that there were many reads that matched the variola virus genome. And the reads were so numerous that they could be assembled into a complete genome, and then the genome compared with the modern day variola viruses of the minor or major clades. And this genome from the 1650s clearly predated the stage when the variola minor and major had split. But if one looked at the gene pattern of that ancient virus from 350 years ago, it had a very similar pattern of gene loss to the modern variolas. So the gene loss must have occurred even earlier. So when was that? Well, a clue to that came last year when a paper by Leeuwenhoek Medal in Science, work from Terry Jones and from Martin Sikora, showed that they could identify variola viruses in bones and teeth from Vikings from many areas in Europe and going back not just hundreds, but thousands of years. And one of these sites that they obtained the DNA from was from Oxford. So the DNA from Viking remains was sequenced, and in 2% of all the samples taken, they found evidence of variola virus. So this indicated that smallpox was more widespread in Northern Europe much earlier than thought hitherto. And the earliest definite case in UK was now 10,002. And that 1002. And that had a special resonance for historians because that is the date of the St. Brice's Day massacre, when Ethelred the Unready decreed that the Danes should be slain because he was fearful that they would overtake his kingdom. And one massacre took place in Oxford, and the massacred young men were cast into a pit. And in 2008, an excavation in St. John's College in Oxford uncovered that pit and found the charred remains of the skeletons of the 35 young men that were shown to be Vikings, and analysis of the DNA from one of those detected variola virus. So if we look at the distribution of the sites that were analyzed and DNA was obtained from all over Europe, you could see that the red ones were where variola virus was found, and there were many of these. And in some cases, enough sequences obtained to assemble complete genomes, and these could then be compared with the modern day variola viruses. So here are the modern ones, and the one from 350 years ago shown in blue. And here are the ancient variolas, which are closer now to the closest animal orthopox viruses to variola, tetrapox and camelpox. And these are diverse, so they didn't derive one from each other in chronological order. They illustrate diversity in smallpox thousands of years ago. And the interesting thing with regard to gene loss is that if we compare the pattern of gene loss in those viruses, there is more genes intact the further you go back in time. And that's illustrated in this slide, where the genes along the bottom are the ones in the modern day variola viruses that are broken and defective. And the open circles indicate where a gene is intact. And as you go further back, you can see that there are more white complete circles than in these modern variolas. And you can plot that with time. So years into the past along the bottom and the number of disrupted genes or absent genes. And you can see that with time, there are fewer broken genes. So it looks like as variola virus was adapting to man, it lost genes, and that might be why it has become a human-specific pathogen. So now to conclude in the last 3 or 4 minutes with a few words about vaccinia virus. So in the 1980s, this virus was developed as an expression vector by 2 groups in the United States, one led by Enzo Paletti and one led by Bernard Moss. And these groups were able to express genes in vaccinia virus. And if these genes were taken from other pathogens, then you could make a hybrid virus that could be used as a vaccine against the organism from which that gene was derived. And this established the principle of using genetically engineered viruses to make live vaccines, a principle that has been applied subsequently to many other viruses and indeed microorganisms. And indeed, in the response to SARS coronavirus 2 epidemic, this technology has been used to make several vaccines, including the one based on adenovirus that came out of Oxford. I'm going to finish with talking about how vaccinia virus is able to evade the innate immune system. And the example I've chosen is how it interferes with interferon. So for those who are not familiar with interferon, this is how this cell protein works to defend against viruses. So if a virus infects a cell, its presence is detected by our sensors called pattern recognition receptors. And that recognition will activate the transcription factors, IRF3 and NF-kappa B, which move into the nucleus and drive transcription of many genes, but including the interferons. The interferons are then produced and secreted from the cell. And once outside the cell, they can bind to interferon receptors on other cells. And that recognition triggers another signaling cascade, the JAK-STAT pathway, that leads to the transcription of interferon-stimulated genes. And the products of those genes are then expressed in the cytoplasm. And these molecules render the cell resistant to subsequent infection by interferon. So these pathways enable viruses to interfere at multiple steps. And indeed, vaccinia virus seems to interfere at every step. So for instance, it can target the pattern recognition receptors or mask the molecules which they would normally use to activate these pathways. It has multiple proteins which will prevent the activation of IRF3 or NF-kappa B, can block transcription from within the nucleus. It even secretes proteins outside the cell that function as decoy interferon receptors that block the ability of interferon to engage the natural interferon receptors on cells. The virus encodes proteins that block the JAK-STAT pathway in the cytoplasm or in the nucleus, and many other proteins that actually target the ISGs themselves and prevent them from functioning. And indeed, you can more or less map out the whole pathway of how interferons are activated and work by studying the mechanisms which this virus has got to interfere with them. And one example of that came just 2 years ago when we discovered that a protein called C6, made by vaccinia, is able to induce the degradation of a host protein, histone deacetylase 4, or HDAC4. And by studying the function of HDAC4, we then deduced that it is needed to recruit STAT2 to the ISRA promoter to drive transcription of the ISGs. And that presumably is why vaccinia virus has evolved a mechanism to degrade that protein. I want to finish with illustrating how abundant these viral inhibitors are. And for the NF-kappa B pathway alone, there are now more than a dozen different intracellular molecules made by vaccinia. These red dots are all vaccinia proteins that individually target the activation of NF-kappa B. And lastly, a new molecule has just been discovered that blocks JAK-STAT signaling, and this has yet to be published. And this work has been done by Callum Talbot Cooper, and Callum studied this protein called 018, that is a small protein, just 60 amino acids, and he showed that it's a very effective inhibitor of interferon induction of interferon-stimulated genes. So in the presence of interferon alpha, these genes are normally produced, but in the presence of 018, their production is shut down. And this is true for interferon alpha or interferon gamma. He showed that 018 would bind to STAT1 and that it would prevent the phosphorylation of STAT1 during infection. So with a virus that makes 018, STAT1 does not become phosphorylated. A virus from which that gene has been deleted, the phosphorylation occurs normally. Together with colleagues in biochemistry, he solved the co-crystal structure of this molecule and showed that the viral protein binds to the SH2 domain of STAT1 in a way that prevents the STAT1 molecule binding to the activated interferon gamma receptor. And lastly, remarkably, this same amino acid sequence is present in paramyxoviruses, very diverse viruses, and their inhibitors function in a very similar way. So a striking example of convergent evolution. And so to conclude, I'd like to thank Callum and the other people who have undertaken this project with 018, and to thank the funders who have supported our labs generously over the years, and to once again thank all the people who've worked in my lab over these years, and to thank the Royal Society for the opportunity to give this lecture to you today. Thank you for your attention.
Geoffrey, thank you so much for an absolutely wonderful lecture. That was a real tour de force of the history of the virus, its evolution, the vaccinology, the pathogenesis, and the extraordinary viral tricks you told us about at the end, which is presumably quite a useful probe for understanding the function of some of those proteins in resisting viruses. So it's a wonderful turnaround that you can use the biology of the virus, even though it's no longer a dangerous disease. So thank you so much. It was absolutely fascinating. The questions are coming in on Slido, so if it's all right with you, I'll just start firing straight away. So up top is from John Macaulay. Do you think that the loss of these genes in variola could lead to a possible loss of a persistent phenotype that might have been present in viruses in human populations when populations were smaller?
That's a good question, John, but I don't think we have evidence to be able to answer that. I mean, pox viruses generally are not persistent because they shut off host protein synthesis, and therefore the cell in which they have infected are destined to die. I mean, human pox viruses like molluscum contagiosum can cause a disease that stays around for quite a long time. But eventually that too is cleared. And traditionally, these viruses are not ones which we think of as causing persistence. And indeed, that's of course why we're able to get rid of the wretched disease by vaccination.
And Dr. Helen Brown asks, do you consider MVA as a promising delivery vector for vaccines in the future?
Yes, the attenuated MVA strain is being widely used in clinical trials against different diseases. I mean, the challenge with a highly attenuated virus like that is to demonstrate sufficient immunogenicity to induce a strong enough immune response against the foreign protein or proteins you're immunizing against while giving the virus. And since it cannot replicate inside our cells, it can only deliver antigen during one cycle of infection, and it can't spread. But yes, it's being tried by many labs around the world and in many different clinical trials. So let's hope so.
Alice Catanzaro asks, is recombination possible between variola strains? And if so, has it been seen?
It's certainly possible. You'd have to have a co-infection of the same cell at the same time by the two different strains of virus. And I suppose the chances of that are pretty low, but it could happen. And indeed, we know that recombination takes place within poxvirus infected cells. Indeed, that's how we first made recombinant vaccinia viruses that expressed foreign genes. And the recombination between different poxvirus genomes had been known much earlier from work from other scientists. So yes, definitely it could happen, but you do have to have both viruses in the same cell at the same time.
That seems to be, I don't see any further questions on Slido. And yeah, I think that may be the end of the ones from the audience. So Geoffrey, I'd like to thank you again very much for an absolutely wonderful Leeuwenhoek lecture.
Thank you, Linda. Thank you for being chair.